All right, today, presentations on spectrophotometry experiments.
Yeah, this one. No, it's not on.
No, it's not.
Turn it on.
You have that little button,
Yeah.
push it on. And if it turns green, you just have to wait about a minute.
Yeah, I don't know. I can start off by saying what our goal was.
I can talk about these stuff.
I can say how we actually did it.
Okay.
Right? That's easy.
And then I can drop it in to close.
Everybody ready? All right, so let's just present over your seniors, second year of science, chemistry, and we're ready to listen to you, and here it comes, all right, I'm getting there. Okay, let's go.
Okay, so for our experiment, we used the spectrophotometer with Skittles. I had found some research on how other people in the past have used spectrophotometers with Skittles. This was the main page that we found the inspiration for it from. So this is an example of the food dyes that are in a lot of different things, but for this experiment we focused on Skittles. And here it shows the wavelength of maximum absorbance. So we figured out that if we could use the Skittles, which Jonah will talk about how we did that, then we could see the graphs and figure out where the maximum absorbance was and then match it up with one of these numbers. And our goal was to figure out what dye was in the Skittles. So that was our goal for the experiment. And then
So how we got these solutions was we got 3 Skittles of each color and 8 milliliters of distilled water, and we just put them in here and then stirred them around for roughly a minute, and we put them into cuvettes.
Then basically how we got each one of our graphs and our maximums, and I just roll through these, but that I would put in a little bit into a cuvette, and then we would just almost take a picture of it, we'd print that one out, go to the next one, do that, and we did that for every single color. And then Jonah and Caroline would figure out the maximum via ruler and then measuring out where it's most stable at the top.
And when we were looking at the graphs, we focused on the peaks, and we know that sometimes, like if Mr. Feeney told us that if the graph was a little bit squiggly in an area, that the reading wasn't as precise there, so we mainly focused on like the straight, more precise line of the graphs to figure out where the maximum absorbance was. So we looked at each graph and found it, and then also found the absorbance. And then by using the research that we found at the beginning, we matched it up. And they didn't match exactly, but we also read that if a graph had 2 peaks, so for example, green, so there's like 2 peaks here and over there, that it could mean that that Skittle had 2 different dyes in it. But we tried to match it up as best as we can and came to the conclusion that those were the proposed dyes in the Skittles.
So did you get a little bit of a match as far as
Yes.
you could name the dye? What was it? What are the dyes in red? What is the red dye number what?
Number 3 is called irafiracin.
Okay.
Yellow 6, number 6 was sunset yellow,
Yellow number 6.
and then yellow number 5 was tartrazine, and then green number 3 was just solid green, and then violet was the last one.
For violet dye, they don't have a special name for that?
No,
We pretty much came up with the conclusion that it was just a mix of a bunch of different dyes.
Mix, okay, good. Okay. All right, what else? Anybody have any questions on this? Did you feel a little more like a scientist? I mean, where you had to look things up, and it's not like the easiest thing in the world. We had to. I like the fact you had to read about it. You said if there were 2 peaks. What did you say, Caroline? If there were 2 peaks?
That meant that the Skittle, like if it was a red Skittle or a red color and it had 2 peaks, that there could have been 2 different food dyes mixed in together.
Everybody see it? Okay. All right. Anybody have a question? very good. Alright, to the next one, next group. Here's a volunteer. You gotta be black, you gotta be black, that's your group, right?
Two Skittle lines in a row.
Well, I didn't necessarily want that, but.
So our presentation is going to sound a lot like the last one because we did the same type of lab with the Skittles and investigating the food dyes that were used within them. So do one of y'all want to talk about what we did with
Did you prepare your dye the same way?
Yeah, we prepared our dye in the same way. 8
Do-
milliliters of distilled water, 3 Skittles, let the color melt off, and then use tweezers to get the Skittles out.
And then we
filter.
drained it actually after we let the color come off. We used filter paper to filter out any candy pieces that would have been left over in the dyes to make sure that nothing would have reflect when we
Okay,
put
hang
it
on
in the
just a minute.
cuvette.
Did you guys do that? Do you have any filtering? Did you have to filter yours or?
We didn't filter, we didn't notice it. Jonah timed it to shake it up so that.
So lots of, did anything settle in the bottom of your test tube? So you had stuff come off.
Yeah, we had there were little flakes of candy pieces that
Okay.
we wanted to make sure didn't go into our
Filter those out.
yeah. So obviously the goal of our lab was to find the maximum absorbance levels of each kind of dye that was used in the Skittles, and then to find what kind of dye it matched with. So this is our data table. So you can see for each one what the maximum absorbance was and what the maximum wavelength was at that, and then we compared it to a chart that had the lists of what the wavelengths were for the dyes, and we tried to see which one it was closest to and then make a guess at what kind of dye was used in the
I'm looking at your numbers. So you had a max absorbance at around 500 and, let's say 36.5?
yes.
And the one they had was 527, which
Mhm.
that could be in that range. And then 629.3, and they had 625, yeah? Wow, that's impressive.
Yeah, and similar to Caroline's group, we had the thing where we had multiple peaks, and so we looked at where it wasn't as squiggly and it kind of smoothed out. That's where we took our reading of maximum absorbance, and that's where we found that it was most accurate to what dye it matched with.
Okay. All right, somebody else go.
What do y'all want to go? You want to talk
Yeah,
about
to the
graphs?
project too. Go ahead and just show maybe show the results or.
These were our results for the graphs.
So orange, tell me about orange. Tell me what that means when you see that graph.
Well, this was the colors that were absorbed, and this was the ones given off. All the way around.
Wait a minute, so what kind of colors, you said the colors absorbed are on the right, but the absorption graph is really low there.
Yeah, it was all the way around.
So which way is it?
This was absorbed and this was given off.
Okay, what color, this was orange, right? So what
Yeah.
kind of colors did it not want to absorb?
Red
And oranges, right?
and orange, yeah.
Okay, go ahead, next one.
And then this is pretty similar. It didn't want to absorb red.
What do you think causes this little jaggedness on there? You're just taking a guess, but the one before that, was it as jagged?
I mean, it was pretty similar.
Similar, some jaggedness, right?
Yeah.
Any ideas about that? I never taught it or anything like that, I just wondered if you had any ideas. Anything in your research talk about that, being jagged?
I didn't see anything.
Okay, all right, go ahead. What's that?
Purple.
What do you think about that?
It also gave off a little bit of red and orange.
What do you mean by giving off?
Like it didn't absorb. the red and orange colors.
Where? Oh, okay. What about that second peak, that little bump there?
I see it gave up those two. I don't know what color that is though.
Well, which one of those wavelengths did you pick, for maximum absorption to identify?
Go back to the chart. I think for this one we used the second little peak. It's keep flipping. There's the first one. We used the second peak, which was closer to like 600. Because there wasn't, kind of like
Right.
Caroline, we came to the conclusion that there's probably a mix of the dyes, but we went with the one that it was closest to, so blue number 2, but it probably had something else with it.
Okay. All right, Jim, tell me the rest of the story.
This is the green Skittle, so it absorbed more of this part of the spectrum, and I think it's blue. I think blue and like the purple. I don't have it on memory.
Uh-huh.
And then it absorbed less of the red and orange, which is a little strange because it's green, but
But there's that second peak there,
yeah,
but it does like that wavelength right there.
yeah, it like does like this wavelength, but everywhere else around it, not
If
so
you
much.
wanna remember the numbers, 400 to 700 is like Roy G. Biv backwards. So 400s are violet, 700s are red. You everybody get that? Okay, go ahead. What's this one?
And this is a yellow Skittle. It really only liked to absorb colors around from 400 to 500, and then every other color after that it did not absorb. So, and that's,
400 to 500 what?
nanometers
Okay.
of wavelength, and then that's the last one.
We found some research, I noticed you flipped through there, any good research you thought was pretty interesting?
I mean, it's basically all that we like talking about what we proved with our, just talking about like what the wavelengths could be and how to test it with the spectrometer.
Uh-huh.
So, yeah.
Yeah, question?
So this might have already been answered, but you guys, what did you say you used to dissolve the dye?
We just put it in water and it kind of just melted off.
Okay.
It was good, I mean, it was good that, water worked really well as a solvent for these. You'll find out later that other people did not use water.
Does sugar not like cause problems?
I think, if you, the people who really did this with real expensive equipment, they probably would have had to do something about that. How do you separate the sugar from the dye? Yeah, that's a good question. That's a very good question. Or I would have to have some sugar water and use that as my blank. If I use the sugar water as my blank, that might, zero out any effects of that. All right, very good. Any questions? That's a good question, Tim. Thanks for doing that. All right. All right, let's have next group. Okay, good, thank you. Yeah, that's fine.
I don't want this one.
You have like the results, or you have a graph.
So we don't have to have it until tomorrow.
I don't know.
Okay, I got you. Okay, thanks.
Oh, it's not here. Okay.
Do you want me to talk about like how we did it? Or you don't have to be objective and goals.
Oh, nice,
Yeah,
nice.
that can start. Okay, so for our lab, we did different types of lettuce. So we did red leaf lettuce, butter lettuce, and then arugula. And Gabby, can you just first talk about how we?
Okay, so how we got each color, we weighed about 1.98 grams of each lettuce, and we put them all into beakers. Then we submerged it with alcohol to the 30 milliliter line and just let it soak while stirring it. And then that's how we got each color of green. We had one that was darker, a darker color, more like purplish.
You wanna show them that?
Yes.
Yes,
How
it
did you
is.
decide then? Why did you decide to use alcohol?
It was, we could preserve it, but we could let it stay overnight. It would take the color off.
Right. So, chlorophyll is more, soluble in, organic, we're gonna get ready to do organic, but, alcohols and things like that, opposed to water. So it would not be good for chlorophyll to be, that soluble in water, because it rains a lot, all the leaves. Can you imagine that? All the rain hitting the leaves and chlorophyll coming out of the leaves, that's not good.
So this was the result of the butterleaf lettuce. It was more of a darker green.
Speak
And
a little bit, more distinct, sorry.
sorry.
A little bit louder.
Oh, this is the butterleaf lettuce that we got. It was more of the darker green color, and we could really see the pigmentation and the
Darker
chlorophyll.
green?
Yeah. And this was the arugula lettuce. It came out as a lime green, more of a little lighter, but still green. And this was the one that was the most different. It was the red leaf lettuce, and it came out on the first day. It was a dark purplish color. But then as we left it out overnight, we saw the color faded and it looked,
So, wait a minute, so when you first put it in there, the dark purple of the leaves, they gave a purplish dye, right?
yes.
You're saying overnight, what happened?
It faded, so it
Really?
seemed, yeah.
Ah,
I think more of the chlorophyll probably seeped out, and so
really?
more of a green color started to
So-
emerge.
Could it be that that chemical degrades when it's not in a cellular, in a cell, environment? Wow, I didn't think about that.
Yes. And then too, like just from the eye, at least, these all look a little different, especially this one looks much darker. But when we look at our graphs, like you can see the butter lettuce here. You can see where the peaks are. This is our arugula. And
What do you
then,
think about those two? If you just look at those two, are they very similar, or would you notice anything different?
yes, I mean, they are mostly pretty similar. There is, where the dips happen, it's a little higher here. Oh, sorry, can't see. A little higher here. But also with the red leaf, it is again, like pretty similar where its
Wow.
peak is. And so you would think that it would be different and more of a purple would come through, but there still is that large chlorophyll presence of the green.
Yeah, I definitely would have thought I would have seen another peak somewhere.
Mhm.
Is this one taken after the color faded away?
No,
This
we
one-
put it back in. It had been a little bit, but it was still like more purple. When we
Yeah.
That's
put it in.
interesting, I think we should have seen that. As a scientist, I would have said, let's do this again.
We
I
did
don't
do
know.
it multiple times too with the different ones, and we
Yeah.
took it out and we put it in again, but
Wow.
it did still show that, so it
And
was
then,
interesting.
In our research, I looked up like why it's kind of purple at first, and like why it's still kind of the same as the other ones. And it said that even though the leaves may appear to have pigments other than green, an abundance of the green pigment chlorophyll is beneath the epidural layer. So like letting it sit for a little bit, and like even though it like appears purple at first, like it's gonna kind of be the same data just because
They
it's
researched
still the
that?
chlorophyll.
Wow.
Mhm.
Well, then that could, that's interesting, because I would have thought, that's very interesting.
And we got the inspiration for this lab because the cautilus leaves are normally done with it from outside, but as you can see, it is not the time where a lot of leaves are outside, and so we decided to do lettuce, which is something that everyone can use. So we wanted to see the different comparisons between the greens to see which one has more
Did
absorption.
you weren't able to do any research on like chlorophyll or anything like that? I mean, with the packet that I gave you did have some absorption, graphs on chlorophyll, but you couldn't match those up at all, could you?
I think we have some maybe in our regular research, but because we didn't have as much time, like
Yeah,
before we
right.
turned
I
it
think
in.
We have-
obviously you didn't get your data till like, late yesterday.
Yeah.
So you can, all right, questions about this? What do you think? That's a little different lab, wasn't it? And what'd you use for your blank?
Just rubbing alcohol.
Rubbing alcohol, okay. Get that. All right, thank you very much. All right, next group.
I think that's us, but Sydney's going to print out.
One more group behind that?
That's, I think that's it.
Okay, EJ. Well, you don't have anything, right? Darn, okay.
I mean, we can go, and we probably won't be able to show results, but we can talk about it.
All right, how about, Sydney's group, can you're gonna wait till, you do that, let's go ahead and do that.
Oh, there she is.
Look at that. Saved by the bell.
Yeah.
Alright, so basically with our experiment, we decided to kind of mess around with the colors that we found in different kind of drinks. And so we used blue Gatorade, a red vitamin water, and this like orange vitamin water. And so when checking the ingredients, we found that this one had no like artificial colorings. This one had like natural vegetable colorings that were like added to it. And this one had like the blue one coloring that's, I think it's in a lot of different kind of things.
So which one had artificial coloring?
This one did.
Oh, the others are not, really?
This one's made out of vegetable coloring, and I don't think this one has any like unnatural coloring. I think it's all pretty natural with the oranges. And so to start off, this is our blue Gatorade chart, which it's really high here, and then it peaks up here.
Where's the peak on the right? What kind of color do you think that is? Remember, 700 is about red, so.
Like orange and yellow.
Yeah.
So that one like to absorb that, right? And then on the left side, it seemed to also want to absorb blue, even though it's blue, right?
Maybe like a dark purple.
Okay, that's fine, okay.
Yeah.
That's interesting.
And so we actually, instead of doing all separate graphs, we kind of combine them. So you can kind of see here, like this is the solid line, but
Ooh, how'd you do that?
just like when you're on the, I did it by accident at first, but then I liked comparing them. But when you're on and you can like enter the different lines, it says like keep or like get rid of. And so I just kept them, and so now you can see them all like pile up, which I think is kind of interesting because now you can see like the absorbance on this side. You can see that this one's absorbing a lot more than this like lower line.
And what's that new one?
This is the pink one with the vegetable colorings.
So it really doesn't like red hardly at all.
Mm-hmm.
But like, so what is that middle? Is that green, yellow?
I guess, yeah, and kind of maybe a little bit of blue.
I really like that putting the superimposing that graph on there.
Mm-hmm.
You might have to show. Eventually, I'd like to know how you did that, but because that's pretty neat. Were you able to put 3 of them on there, or just 2?
Yeah, here's the third one, which I think is probably the most interesting.
Wow, this is very, very good. I'm
Yeah.
impressed.
So now I thought like we weren't totally sure like what we were gonna get out of it, but I think it's super interesting that like the one without any artificial dye like has the highest absorbance, and then it like continues going down.
Violet blue.
Mm-hmm. Yeah, so I thought that was very interesting.
Yeah, and it doesn't show. I mean, that's the highest absorbance, but no other little peaks anywhere.
Mm-hmm. It's just like it's almost linear.
That's interesting. Huh. I've never seen an absorption graph that smooth without little peaks and things. So that was interesting. All right, now let's have somebody else talk.
Someone can talk about like how we did it. I guess it's pretty simple.
Alright, so what we did basically is we just took samples and poured these in the cuvettes, and then we just found, I think we put the blue one in first.
Yeah.
Put the blue one in first, and then we just went down the line to see which ones it would absorb, and then we just graphed them as we went. I mean, it wasn't the same as the dilution lab because we didn't have to find the highest concentration first and put that one in. We were able to just put each of the separate ones in.
Yep. Okay. Did anybody think about doing anything besides, you just said having more, you said 3, right?
Yeah.
Okay. Did
Well,
it make you want to look at some other ones, or?
It was kind of, we weren't totally sure the effects of like, we made sure to get 3 that weren't carbonated because we weren't sure like how the air bubbles like within the cuvette would work, if that would like mess up the light refraction.
If we did have a carbonated, we might have to have it set out overnight for 48 hours
Yeah,
or
so
2
it's
nights,
completely flat.
to get rid of the carbonation. Very good. All right, anybody else? Who else want to talk? Anybody else have anything about research or anything like that?
Well, we found some research about it because we've seen like this experiment had kind of been done a couple different times before, but a lot of them had to do with like using different types of Gatorade. And so like in Gatorade, they all have like, pretty much all of them have artificial dyes. So like our experiment kind of was a bit different since like we had vitamin water, which uses more like natural dyes. So that was pretty interesting to find like It kind of explains why like the graphs don't look as similar, because like with Gatorade, like even though with the different colors, like they'll peak at different points, they still kind of have that like one peak. But then we see like with the yellow vitamin water, like it kind of just doesn't even have a peak. So that was pretty interesting finding the research.
So, in your opinion, the one with no artificial coloring, wait a minute, two of them had no artificial coloring, right?
Yeah, one of them had vegetable, like natural vegetable colorings that were added to it, but one just didn't have any.
But even if you get it from a plant, natural coloring, it's still a dye, isn't it?
Yeah.
So, you didn't notice anything about artificial versus natural dye, did you, or any
I
conclusion
mean,
on that?
the natural dye, I think is the one that had the natural dye is the one that had the straighter lines.
It is?
Oh, it's the middle. It's the middle wavelength. It's the middle one. The blue is the lowest, which has the artificial.
Can you point it out just real quickly again, what's the smooth line?
This is the blue.
The yellow.
This is the pink, and this is the yellow.
Yellow, okay, all right, thank you. All right, what do you think, questions on that, you like that? All right, thank you very much. And is that it for today? All right, is that it? So DJ's not here, right? Okay. I could have you go up there, but you don't have anything to show, and so I don't think I wanna do that, so maybe you can meet me sometime for about 10 minutes, and do the presentation for me, is that all right? All right, what I'd like to do now, in the rest of the period, this has really helped me a lot. We were in the lab before Christmas, remember that? We need to clean up. So what I'd like to do is, as much as you can, clean up. Now we have, back here, if you go through that door, there's a sink there, there's even, a little bit of soap. If you need to get a beaker full of soap, water, you can do that, but also anything that's in the drying rack,